Hello everyone, my name is Dr. Diana, and I am going to help you in understanding the role of loop of Henle and distal nephron in concentration of urine. In this section, we will learn about the role of loop of Henle in concentration of urine. So, let's start. This is Bowman capsule. This is proximal convoluted tubule. This is thick descending limb. This is thin descending limb. And then there is loop of Henle. Then thin ascending limb. Then there is thick ascending limb. This is distal convolute tubule. And this is collecting duct. Now, these are the processes involved in concentration of urine. I know you are getting confused, but there is nothing to worry about. I am here to simplify it for you. The loop of Henle and distal nephron allow urine to be concentrated through the creation of a high osmolarity in the medulla, which drives the reabsorption of water from the collecting ducts. The distal nephron also regulates potassium and calcium excretion and acid-base status. Let's break them and study one by one with the help of in-depth mechanisms. But before we start them, let me point out some key features for you. The thin descending limb is permeable to water, but impermeable to urea, whereas the ascending limb is impermeable to water, but permeable to urea. The distal tubule and cortical collecting duct are impermeable to urea. But remember that the medullary collecting duct is relatively permeable to urea. They are also impermeable to water, except in the presence of antidiuretic hormone, or vasopressin, which is the other name of that antidiuretic hormone. So it means, in the presence of antidiuretic hormone, the distal tubule and cortical collecting duct are permeable to water. Let's move into detailed discussion of loop of Henle in concentration of urine. Below this boundary, there is medulla, while above this boundary, there is cortex. This is loop of Henle, descending limb of loop of Henle, and ascending limb of loop of Henle lie in medulla. This is thick descending limb. This is thin descending limb. Then there is hairpin bend of loop of Henle. Then this is thin ascending limb. And then this is thick ascending limb. Thin descending limb is permeable to water, but impermeable to urea, whereas ascending limb is impermeable to water, but permeable to urea. From proximal convoluted tubule, fluid entering the descending limb of the loop of Henle is isotonic with plasma, which is 300 milliosmol per liter. The generation of high osmolarity in the medulla depends on the differential permeabilities to water and solutes in different regions, the active transport of ions in the thick ascending limb, and the countercurrent multiplier. The thin descending limb is permeable to water, but impermeable to urea, whereas the ascending limb is impermeable to water, but permeable to urea. This has already been discussed. When the fluid passes from proximal convoluted tubule into the thick descending segment, water is reabsorbed from tubule into outer medullary interstitium by means of osmosis. It is due to the increased osmolarity in the medullary interstitium, which is outside the thick descending tubule. The osmolarity of the fluid inside this segment is between 450 and 600 milliosmol per liter. That means the fluid is slightly hypertonic to plasma. As the thin descending segment of Henle loop passes through the inner medullary interstitium, which is increasingly hypertonic, more water is reabsorbed. This segment is highly permeable to water, and so the osmolarity of tubular fluid becomes equal to that of the surrounding medullary interstitium. In the short loops of cortical nephrons, the osmolarity of fluid at the hairpin bend of loop becomes 600 milliosmol per liter, and in the long loops of juxtamedullary nephrons, at the hairpin bend, the osmolarity is 1,200 milliosmol per liter. Thus, in this segment, the fluid is hypertonic to plasma. When the thin ascending segment of the loop ascends upwards through the medullary region, osmolarity decreases gradually. Due to concentration gradient, sodium chloride diffuses out of tubular fluid, and osmolarity decreases to 400 milliosmol per liter. The fluid in this segment is slightly hypertonic to plasma. This segment is impermeable to water, but there is active reabsorption of sodium and chloride from this. Reabsorption of sodium decreases the osmolarity of tubular fluid to a greater extent. The osmolarity is between 200 and 150 milliosmol per liter. The fluid inside becomes hypotonic to plasma. This fluid then moves to the distal convoluted tubule that will be discussed in next section. This is the tubular osmolality. But what about the interstitial osmolality? Well, this is same as that of tubular osmolality. But how the osmolality of interstitial fluid becomes same to tubular fluid? This can be justified after the discussion of the thick ascending limb in detail. These are tubular cells. This is interstitial fluid, while this is the lumen. The thick ascending limb actively reabsorbs sodium and chloride from the tubular fluid by means of apical sodium-potassium chloride cotransporters. Sodium is primarily transported across the basolateral membrane by sodium pumps, and some by sodium bicarbonate cotransport, and chloride by diffusion. Potassium leaks back into the lumen through apical potassium channel, called as renal outer medullary potassium channel, creating a positive charge that drives the reabsorption of cations, such as sodium, potassium, calcium, and magnesium, through paracellular pathways. As you can see, sodium and chloride are moving in interstitium from tubular cells of ascending limb of loop of Henle, so that this increased the osmolality of medullary interstitium. Main reason for the hyperosmolarity of medullary interstitial fluid is the active reabsorption of sodium chloride and other solutes from ascending limb of Henle loop into the medullary interstitium. These solutes accumulate in the medullary interstitium and increase the osmolarity. So this hyperosmolality of interstitial fluid is decreased by two ways. Due to the concentration gradient, the sodium and chlorine ions diffuse from medullary interstitium into the descending limb of Henle loop and reach the ascending limb again through hairpin bend. Thus, the sodium and chlorine ions are repeatedly recirculated between the descending limb and ascending limb of Henle loop through medullary interstitial fluid, leaving a small portion to be excreted in the urine. And second way is by that. When the fluid passes from proximal convoluted tubule into the thick descending segment, water is reabsorbed from tubule into outer medullary interstitium by means of osmosis. And we know that osmosis is movement of water from region of lower concentration towards the higher concentration. As hyperosmolality of interstitial fluid causes water movement out of tubule into medullary interstitium. Summary of countercurrent multiplier is that the increased interstitial osmolality causes water to diffuse out of the descending limb, and some sodium and chloride to diffuse in, concentrating the tubular fluid. As this concentrated fluid descends, it travels in the opposite direction to fluid returning from the still higher osmolality regions of the deep medulla. This countercurrent arrangement creates an osmotic gradient, causing sodium and chloride to diffuse out of the ascending limb and diluting the ascending fluid, and water to diffuse out of the descending limb, further concentrating the descending fluid. This effect is potentiated by the fact that the ascending limb is impermeable to water, but highly permeable to sodium and chloride, and also by the recycling of urea between the collecting ducts and ascending limb, which makes an important contribution to urine concentration that will be explained in detail in next section. At the tip of the loop of Henle, the interstitial fluid can reach an osmolality of 1200 milliosmol per liter, due to equal parts to sodium chloride and urea. The blood supply to the medulla is prevented from dissipating the osmotic gradient between the cortex and medulla by the countercurrent exchanger arrangement of the vasa recta capillaries. Now let's discuss the vasa recta. Vasa recta functions as countercurrent exchanger. It is responsible for the maintenance of medullary gradient, which is developed by countercurrent multiplier. Vasa recta acts like countercurrent exchanger. Because of its position, it is also U-shaped tubule with a descending limb, hairpin bend, and ascending limb. Vasa recta runs parallel to loop of Henle. Its descending limb runs along the ascending limb of Henle loop, and its ascending limb runs along with descending limb of Henle loop. Thus, sodium chloride is exchanged for water between the ascending and descending limbs of vasa recta. Hence, this system is called as countercurrent exchanger. It should be noted that oxygen and carbon dioxide are also conserved, so that in the deep medulla, partial pressure of oxygen is low, and partial pressure of carbon dioxide is high. The vasa recta also removes water reabsorbed from the loop of Henle and medullary collecting ducts. This is the flow of blood. So the conclusion is that loop of Henle functions as countercurrent multiplier. It is responsible for development of hyperosmolarity of medullary interstitial fluid and medullary gradient. But vasa recta functions as countercurrent exchanger. It is responsible for the maintenance of medullary gradient, which is developed by countercurrent multiplier. This was all about this section. In our next section, we will talk about the role of the distal tubule and collecting duct in concentration of urine.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.